Yes, thank you very much for this opportunity to speak. And yeah, I would like to apologize for my lack of Norwegian. I'm not yet at the level to be able to deliver this in understandable Norwegian. So I will stick with my native English, and I hope that many of you are still able to understand well. I will try and speak slowly, which is often yeah, something that I struggle with as a native English speaker. So I will try my best to enhance understandability today. Yes, so I'm going to be presenting some of the results that are emerging out of an EU project that we coordinate at NIVA, that is Papillon. So a little bit first about what is Papillon. It stands for Plastic in Agricultural Production, Impacts, Life Cycles and Long-Term Sustainability. It's a Horizon 2020 Research and Innovation Action funded by the EU. It runs for a period of 4 years, and as you can see from the slide, we're just coming up to the last few months of the project now, and we're starting to get all of these results coming out in quick succession now. So the results I'm presenting today are sort of fresh off the press, and there's sort of more results coming out next week, the week after that, the week after that. And so I'll give a list at the end of all of the results that are still to come as a bit of a teaser for some of the results that you can expect from the Papillon project. We're 20 partners across 12 countries, and NIVA in Norway are the coordinators of the project. So we manage the project, but also we are one of the main analytical laboratories in the project, and we also have a task for a socioeconomic cost-benefit analysis as well. But I'll be presenting results from across many of the partners. It's a very collaborative project, so many of these results I'm presenting today come from several different countries and different research groups within the Papillon Consortium. So the primary objective of the Papillon project is to fill knowledge gaps on the sources, behaviour and long-term ecological and socioeconomic impacts of micro and nanoplastics from agricultural plastics in European soils, and to provide the scientific background to enable policy, agricultural and industrial innovation towards sustainable farm production systems. And we articulate this through research that occurs at the sort of field scale, but also very much at the farm scale as well. Some of the key tasks that will be kind of part of the Papillon project. This includes an atlas of agricultural plastic use, practices around this plastic use, and potential micro and nanoplastic releases. This sort of feeds into knowledge on the sustainable use of agricultural plastics, how they can potentially break down to form micro nanoplastics, and how we can kind of help prevent this process. We've also developed some environmentally relevant reference materials to underpin scientific testing, so that all of the results that come out of the project are actually using micro nanoplastics generated from relevant source materials, that is agricultural plastics. We've also developed a number of harmonized analytical methods for detecting microplastics and also plastic additives in soils. And this will feed into a large program of scientific testing where we will have a risk assessment framework for micro nanoplastics in soils. And all of this will then be combined together at the end in a task that is currently ongoing, which is the socioeconomic cost-benefit analysis for agricultural plastics. The Papillon approach, how we kind of bring all of this together in terms of our scientific testing. We take these test materials that we have developed, we take the novel methods that we have harmonized, optimized, developed in the project, and then we run these through a series of experiments, starting from very small scale laboratory studies where we can test very specific mechanisms or processes. And then we slowly expand the spatial scale of these experiments to mesocosm experiments and field plot experiments and a large scale field monitoring, where we go from having a very high environmental control, but a small sort of spatial scale, up to a large spatial scale that is very environmentally relevant. And so we can see how these mechanisms or processes or effects that we see at the small scale then play out as we add in more and more complexity. So I will present some of the results from some of the high environmental control studies in terms of effects, and then also a few at the other end of the scale in terms of some European spatial survey results as well. So the content of today's presentation, I will start off with some preliminary results from some European soil monitoring, which includes some monitoring that took place in Norway. Then I'll move on to talk about some emerging evidence about the field degradation of biodegradable mulching film from a field burial test across different parts of Europe. And then I'll end by just quickly summarising some of the emerging results on the impacts of plastic pollution on both plants and soil animals. So moving promptly on to part one, I will get straight into this European monitoring study. So we undertook sampling of 73 agricultural fields across 7 countries in Europe. And one of those countries, as I mentioned, was indeed Norway. Within each country, we selected fields for inclusion based on 4 source categories. And these were selected based on their potential to input microplastic to soil environments. So one of those was the use of agricultural plastics. For this, we prioritized only sort of the conventional non-degradable polymers so that we could have a little bit more statistical control over the results instead of adding lots of different types of plastic. So this is just the non-degradable polymers. But different agricultural plastic applications. So the use of mulching films, low tunnel systems, greenhouses, irrigation systems, etc., etc. The second category relates to the application of sewage sludge, where there's a lot of literature emerging that this sewage sludge can be enriched with microplastics that come through the wastewater treatment plant from a variety of sources, and then can tend to be kind of enriched in this solid phase, which is then used as a soil conditioner and fertilizer in many countries. The third category was the application of different types of compost or digestate products. These vary very much across Europe, but also within regions and countries in some cases in terms of what actually goes into these composts or digestates. So we kept a definition fairly broad for this category, especially because these words don't sort of mean the same in different countries. But it was generally compost or digestate products that were suspected to be contaminated by microplastics based on the waste streams that led into them. And then finally, the fourth category was our kind of reference sites, and that is where there is no history of plastic exposure from those other 3 sources for at least the last 10 years. And on each field, we had a very strict sort of sampling design. So every field sampled in all of these different countries was sampled in exactly the same way, so that we can then have these harmonized results. We can compare between the different fields across the different countries. And you can see here there were actually a lot of different things that we sampled. I'll just be presenting the microplastic results here today. So straight on to those results. I will just present some of these ranges and averages. These are very fresh results. They're currently going through the publication phase now, and we're also doing some final quality checks on these. So these are kind of these preliminary results that are now emerging. So what we can see here are the ranges for the whole of Europe across all of these different source categories, where the levels of microplastics in soils range from around 2,000 up to just over 400,000 microplastics per kilogram dry weight of soil. When we look at the average, it's around 60,000 microplastics per kilogram of soil, and the median was a little bit lower at around 26,500 microplastics per kilogram. These are generally higher than the levels that have been reported so far for Europe, and that's because we were able to go down to quite a small particle size. So other studies that have reported lower concentrations were missing some of these small particles that we were able to detect in this study, and that's why we're seeing such high numbers here. If I look at the different source categories, you can see how these break down the range of fields across the whole of Europe. So for agricultural plastics, it was around 3,000 to just over 400,000. For sewage sludge, the lowest was around 16,000 up to nearly 200,000. Compost and digestate, that was quite similar to the agricultural plastics. And then the reference fields, again, around 2,000 up to just over 300,000 microplastics per kilogram. I guess you're pretty interested to hear how the results fare for Norway, so I can give the range here. So Norway, the minimum concentration we saw was just under 5,000 microplastics per kilogram dry weight of soil, and we went up to the maximum we saw was 67,000. So we're actually just over the average for Europe. So as you can sort of extrapolate from that, there are many other countries in Europe that show much higher levels of microplastic contamination. So we are relatively low in terms of those levels when we compare to the region. But something that is notable is that macroplastic, that is the large pieces of plastic, were also surveyed. And despite these sort of low levels of microplastic relative to Europe, we did have the second highest macroplastic contamination across the whole of Europe on one of those fields. So you can see that these results do vary a little bit depending on what component of plastic pollution you are looking at. So what we can see from those results is there's not really a clear dominant input from any single source category. A lot of those numbers overlap, right? When we drill down on agricultural plastics, which obviously the basis of this forum, we're pretty interested here today. In the fields where agricultural plastics were used, those agricultural plastics that had been applied contributed between 14 and 40% of the microplastic that we detected. And because we see that there is this then at least 60% of the piece of cake missing, and we also saw that these levels from the different source categories overlapped, we can take from that that there are additional as yet undefined sources of plastic to soil. So these include sources that we didn't look for in the study explicitly. Amongst the potential options, atmospheric deposition, we are starting to see may represent a pretty important contribution to soil microplastic levels. We've been able to tease some of this out of the results that we're analysing right now. But something that can hopefully leave some concern about this microplastic that is potentially raining down from the sky that we have little control over. The results are starting to show that Norway and the Northern European countries may actually receive lower inputs of this atmospheric deposition of microplastics than Southern Europe. So we are a little less affected from that atmospheric deposition than other parts of Europe. So what's coming next? Well, we have a full suite of results looking at different types of plastic pollution and associated chemical pollution across all these fields, as well as understanding potential effects on earthworm abundance and biodiversity, microorganism communities, and also soil properties. And something that we're doing right now is to develop a model to translate these particle counts that I mentioned before, these sort of numbers of microplastics per kilogram of soil, into some mass-based estimates. And this feeds into a systematic literature review that we've performed on the occurrence of plastic contamination in global soils, where we've specifically focused on the role of agricultural plastics as a source. We see that the levels generally fall between 0.0001 and 0.01% of soil. And so we will map these monitoring results on top of that literature review of these global soil levels that have been detected and reported in published studies so far. But what we're starting to see is that microplastics are still sort of starting to emerge as one of the most abundant anthropogenic components in soils. So we are seeing that these microplastics do represent quite a large relative proportion of soils when we take those other different anthropogenic inputs. But leading on from that, I will talk a little bit later about what does that mean for our soil ecosystems, and is that something to be concerned about? In the meantime, I'm going to fill a little bit of a gap left in that monitoring survey where we did not specifically look at any fields using biodegradable plastics. So instead, to have a look at the role of biodegradable plastics as agricultural plastics in terms of a potential source of micro nanoplastics or other forms of plastic and chemical pollution to soils, we performed an in-field degradation burial test of these mulching films in different regions of Europe. So we took intact, pristine and artificially aged samples of biodegradable mulching film, and we buried these in soils in 3 locations in Europe. We had Finland representing this northern European environment, we had Italy, and then we had southern Greece. And the aim was to test the biodegradation rates in these different climates and soil types, and specifically under field conditions. So with just sort of the weather patterns that were happening in the field with all of the other complex processes going on. And this was to test the real world variability that exists around the different parameters that are tightly controlled in soil biodegradability standard tests. So we wanted to know what actually happens in real world settings compared to these standard tests that are used to confirm the biodegradability of these agricultural plastics in soil conditions. A brief summary of the results. The first full results are now available for Italy, and we're finalising the results for Greece and Finland, where I guess Finland is most of interest to you guys. In Italy, we saw 50 to 65% surface loss of the samples after 478 days. And this represents this like disintegration of plastic. So it's just what was able to be retrieved from these grids that held the films in place. Chemical testing of the plastic residues that were left over this time revealed that the starch component degrades first. So what actually happens first is the starch is biodegrading and it helps to fragment the plastic. But what it entails in the short term is this enrichment of the polymeric component relative to that starch component. So as the starch is biodegraded and being lost, we end up with more and more of the polymer left in the soil during this biodegradation process. And as a result, we see some components of this polyester component, this polymer component, which is terephthalic acid, that tends to be enriched in the soil during that degradation process. And something else that's relevant, we see that there's this disintegration. We see that there's a sort of loss of this intact film in the figure in the top corner there. But those sort of purple spaces that are left in between, this doesn't represent a full, complete biodegradation of those plastics in that time. What is also in amongst those spaces are microplastics that were then lost from these grids and inputted to the soil surrounding this burial test that then were too small to be retrieved when the grids were lifted out. So there's also this kind of temporary input of microplastics to the surrounding area as part of this biodegradation process over time. Some of the things that were noted as part of all three experiments is that degradation is quite affected by moisture content. There's a bit of a Goldilocks effect on this. So too much water, and we don't see much biodegradation at all. It introduces these anaerobic conditions that prevent some of the microorganisms from actually biodegrading these with these mulching films, but also too little. That can also halt biodegradation as well. So during periods of drought, the biodegradation is noticeably reduced. So what we can see from the results is that the degradation of these biodegradable mulching films does actually occur within the 2-year timeframe that's set by certification standards. But this is not in terms of 2 years of calendar days. This is in terms of thermal days. So this is the number of days where the biodegradation temperature threshold was met. So in Greece, for example, almost every day is in fact one of these thermal days, and so the 2-year time frame seems to roughly apply. But in Finland, soil temperatures were actually below 5 degrees for, on average, 6 months of the year, and 5 degrees has been noted as the point at which there is no degradation at all. So we can see that actually the degradation time is significantly increased by these low soil temperatures that persist for large parts of the year. So what we can take from that is that degradation is also expected to take much longer under Norwegian field conditions. Okay, moving now on to part 3, I'll talk a little bit about a summary of the emerging results on the impacts of plastic pollution on plants and soil fauna. Starting with effects on plants. So at the beginning of the project, there was a large literature review that brought together all of the literature that had tested microplastics or nanoplastics on different plant species with different types of experiments. The results of all of these brought together in this meta-analysis shows that there is pretty unanimous confirmation that micronanoplastics can cause negative effects, and this includes some effects that occur at environmentally relevant concentrations. Many of these studies actually tested much higher concentrations that we don't see in sort of Norwegian agricultural settings or even in global agricultural settings, to kind of test these really extreme cases and to see at what concentrations we do see any sort of large-scale negative effects. But several studies did include concentrations that we do see in the fields in Norway, in the world as well, and saw some negative effects at those levels. These effects include changes and reduction in germination and growth. And also stress indicators and stress responses are fairly consistently upregulated. However, these results that were all sort of pulled together in this meta-analysis, they mostly relate to studies that test one microplastic type and often on one plant species, and usually in lab conditions, for example, in a hydroponic system. So what we need now is testing of, for example, multiple species with the exact same microplastic test materials, including testing in, for example, mesocosms or field experiments to try and understand that level of complexity, and also to see how these effects play out when you've got sort of these same variables in place. So that is what we have been busy doing over the last few years. Something that we've seen when we've applied this same microplastic type to a suite of different species is that the effects that were observed in this meta-analysis, they can be species dependent. For example, growth and germination tends to be more affected in lettuce and carrots rather than monocots like wheat and barley. And also these biochemical stress indicators that we mentioned, these were upregulated to a greater degree in lettuce and wheat. So we saw these differences in these different species where we can't necessarily apply these kind of broad negative effects that are talked about to every single species that is tested. We've also tested different microplastic types, specifically conventional and biodegradable microplastics derived from plastic mulching films. And when we test this, we see that the biodegradable microplastics exert more adverse impacts than the conventional plastics. An example of this is we see negative effects on chlorophyll content, namely a reduction in chlorophyll content. We see an increase in oxidative stress, and we see a reduction in germination and growth parameters. Again, this tends to be a little bit more enhanced for the dicot species, for example, lettuce and carrots, than it is for those monocots, such as wheat and barley. So we start to see that the different microplastic types that these different crops can be exposed to in the field environments can be affected differently. So again, we can't have this sort of blanket assumption to all of these different microplastic types in all of these different crops. What's next is testing this effect under real-world conditions in a long-term field experiment. So results that will be available in about a month or so will show how these effects play out in a study that uses both conventional and biodegradable microplastics that have been applied to experimental fields in 3 locations in Europe. And we've then sown crops and maintained these for a period of 2 years. So we see how these effects actually play out at real-world conditions and across relevant timescales. So these are results that are still to come. A slightly less clear-cut picture are the effects on soil fauna. So there are several organism groups, for example, springtails, where even across multiple generations and testing lots of different microplastic types, no adverse effects were observed. We do see some potential effects on some species, for example, at high concentrations, there is oxidative stress observed for earthworms, and there's some interesting patterns in terms of growth occurring in response to different microplastic types, whether those are conventional or biodegradable, that don't necessarily translate into adverse effects, but show that there's something going on. Again, we have more results to come with this. We have multi-generational testing of several more species, including different types of earthworms, encotrade worms, nematodes, ants, woodlice, beetles and snails. So we are testing across a wide suite of different soil fauna. So again, we can see how these effects play out. And this is taking place at the lab scale, these complex mesocosm scales, and also at the field scale as well. So we can see how these actually play out across different spatial scales. And then the ambition is to bring all of this together. So the results from this large-scale multi-year field study, this will integrate effects on plants, soil fauna, and also soil properties, including microorganism communities as part of that. We're starting to see from these results that are emerging like in these days that we're experiencing now, and we start to see some important changes in soil properties that might be mediating some of the effects that we see. This includes some significant short-term changes in soil pH, especially related to biodegradable mulching film use, where there is a reduction in pH that occurs within a matter of weeks. But then also long-term changes in nutrient cycles, in particular nitrogen cycling, and these are occurring at environmentally relevant microplastic concentrations. So these are occurring at concentrations that fit right in the middle and the lower end of that global microplastic occurrence graph that I showed earlier on. So these are effects that could be occurring today in our fields. So these are the results that we will come out with in just a matter of months. So just to whet your appetite for what else you can expect from the Papillon project, we will soon be releasing the full suite of results from that European monitoring survey, including obviously the Norwegian part, which we will then communicate in Norwegian, so that then it's very accessible to Norwegian farmers, and we will obviously distribute that widely, so you can then see the results of that monitoring survey across the whole of Europe and the specific results for Norway and how that fits into regional averages and regional trends. We'll also then, of course, be having the information on the biodegradation rates for those mulching films, the biodegradable mulching films in Finland, which obviously has a better translation to Norway than that Italy case study. We will then have, as I mentioned, the results from this wide range of multi-generational single species tests on plants and soil fauna that demonstrate a range and sort of variability of microplastic effects. There will also be the results from these large-scale and field studies that test these sort of effects that we observe at the laboratory scale under these more environmentally relevant conditions. There will also be this European Atlas of agricultural plastic use and waste handling, and this will include the results of this mapping effort that's been undertaken from Norway. And finally, some work that's very much in progress as we speak, information about sustainable practices around agricultural plastic use, including a socioeconomic cost-benefit analysis. So we can really understand when we factor in all of these different effects that we see, what is the sort of socioeconomic cost-benefit of actually using these agricultural plastics, and how does this play out in terms of long-term ecological sustainability? So this is information that is still to come. And with that, I'd like to thank you for listening. And if there's any questions, you can also drop me an email. I've just put my email address there as well. Thank you very much.
Thank you, Rachel. It's a very big research project, and many fields to be discovered and investigated. And of course, everyone wants to know, does this apply to me? So what do you think, how representative are these fields you have been choosing, the salad production and the grain production? Do you think how many farmers will be affected by this, since you have been doing this mapping? Can you say something about that?
Yeah, I think it's hard to come up with concrete numbers just yet because we're still sort of obviously working with those. But I do think these results are quite relevant in terms of the variety of fields that we included in the Norwegian monitoring, and how those have then linked with that mapping exercise. So we have some of these are connected. We've been working together with those farmers, and the results then there will be obviously like a translation to what this means for real world effects. A lot of those field results are relating to barley production, which is obviously quite relevant also for Norway. So these effects are going to be on a crop that is also sown in Norway. So we can translate some of the Finnish case to the Norwegian case as well, because there's a little, obviously it's a lot more similar, Finland and Norway versus Norway and Greece, for example. So we can start to sort of tease out some of those effects and actually apply those to the Norwegian level and make sure that that is representative, and explain some of the nuances of that as well. So we don't want to be alarming with the results. We want to communicate them in an effective way. So we'll sort of communicate how those fit in, and what we can really take away from those in terms of the message.
Does crop rotation has been investigated?
No, we didn't include crop rotation at this stage. So the intention is to look for follow-up funding to this. These large-scale field experiments where we could potentially explore crop rotation, they're obviously quite expensive to run and they take a long time. So in this project, we could just do this one. So we then tested this sort of first phase of complexity, and then the ambition would be to continue those in further funded projects to start to look at some of these different processes. So the fields where these microplastics were applied, they will be maintained. The ambition is for them to be maintained. Also to make the most out of these fields where we have added these microplastics, so that we're not then just adding microplastics and sort of walking away and leaving this contaminated soil. We want to get the most benefit out of that, and then we will also clean up those soils after the experiments are completed.
I was just thinking about like applying these products from the biogas production, so which will leave some microplastic on the soil, and if people are taking over the field and suddenly growing something that will not grow anymore as expected, so that might many perspective we have to consider, like using any kind of product that contains plastic or using plastic directly on the fields.
So yeah, and we have a follow-up study already taking place in Norway where we're sampling. We've already sampled several more fields than were included in this European monitoring, and that includes some where we've got this biogas digestate in Norway. So we will also come with more monitoring data a little later on. We're still analysing those results, so we're still in the lab with that. But we will have even more data, including a wider variety of farms and different source inputs that are very, specifically Norwegian. So we will have more data on that soon as well.
All right.